Okay, everybody, today I'm going to teach you about protein synthesis. So what is protein synthesis? It means that you're going to make proteins from DNA. So we've looked at the genetic code, and you know that you have, I'll just pull that up in front of me, that you have these things called codons, 3 nitrogenous bases in a row code for an amino acid. So how do we make these amino acids? We have to open up the DNA molecule and read it, and then get those instructions on which amino acids we're supposed to put together. Okay, and once we put those together through peptide bonds, and the peptide or the polypeptide then takes on its secondary or tertiary or quaternary structure, we have a protein. So protein synthesis occurs in 2 stages. Okay, the first one is transcription. All right, so transcription means that we're going to change the language of DNA into an RNA copy. All right, so this is my second take on the video. So if you're wondering why this is already filled out, that's why. I'm going to give you time, and I think you can pause the video as well. So transcription means we're going to change DNA into mRNA. All right, what does mRNA stand for? It stands for messenger RNA. Okay, transcription occurs in 3 phases: initiation, elongation, and termination. So we're going to see that with part 2 also, which is called translation. So what I'm going to do today is I'm going to make 2 videos for you. I'm going to deal with transcription in one video and translation in another. If you were sitting in front of me in the classroom, I would definitely be teaching all of this in one day. So it's totally up to you. I'm going to give you this information for Friday and for Monday's lesson. You can watch both videos in a row to see how one flows into the other, or you can take it into 2 lessons, whatever is most convenient for you guys, because I know that right now things are not normal and many of you still have jobs that you're going to. So we have 3 phases in transcription, okay? And I probably should have said A, B, C, but I wrote these down first before writing that down. So we've got initiation, we've got elongation, and we have termination, okay? And as we go through transcribing the DNA molecule into an mRNA molecule, I'm going to touch on each of these. But I just thought I'd put a little bit of information in here for you so that you've got an idea of what actually happens in each stage, and when the stage begins and when it ends. So in initiation, we're going to have RNA polymerase looking for a TATA box. Okay, so that says TATA. And the TATA box is something we find in eukaryotes. So I'm going to use the TATA box today. If we were copying prokaryotic DNA, we'd be looking for TAT, AAT. Okay, so when I quiz you or give you anything else, I mean, you're going to have these notes in front of you anyway, but if it was a normal situation, I would put the TATA box on the quiz, and you'd have to find it and say, okay, that's my promoter. So the promoter basically says this. It says after the TATA box, that's when we start to make our copy. Okay, so we're not going to copy the TATA box. We're going to look after whatever letter comes after the TATA box, and that's when we start to make an mRNA copy of DNA. And then we've got elongation. We're going to have the molecule elongated. So what are we elongating? We are elongating mRNA. Okay, so mRNA is elongated. What does mRNA stand for? It's our messenger. So mRNA is our messenger. Why do we need mRNA? Because DNA can't leave the nucleus. It's much too big. So because it's not able to leave the nucleus, it's a big double helix structure, we make RNA copies of it. And the RNA copy of the DNA is called the messenger copy. All right, you're going to see 3 different forms of RNA in these lessons. The first one we're dealing with is mRNA, standing for messenger RNA. It takes the DNA message outside of the nucleus, all right, in an RNA format. And then finally, we're going to be looking at, okay, when do we stop copying this DNA sequence? And so what we're going to be looking for is a series of A's, many A's. I'm condensing it into 5. There would be many, many of them, okay? But we don't have time or space to write down 50 A's in a row. So we're going to be looking for a series of A's on the template strand. Once we encounter that, then those guys are transcribed as U, because if we're changing the DNA into RNA, we know that the partner for A is a T on DNA, but for mRNA or for RNA, it's a U. So we will transcribe this into a U form. That'll make more sense in a minute once I get started. all right? And then what I want you to know is that nuclear proteins bind to this UUUUU, which is on the mRNA, and transcription is done, okay? So one more point of emphasis before we get started. When we copy DNA, we copy the entire molecule because we want all our cells to have the exact same DNA, all right? When we're making proteins, think about this. Your DNA is a code for how to make every protein that you need in your body, all right? So if you're interested in making hemoglobin, you have no interest in making keratin, the protein in your hair. So it would be really wasteful if you're just interested in making more and more hemoglobin to make keratin. So what point am I making here? What I'm trying to tell you is that when you're going through protein synthesis, you're only opening up a very small portion of your DNA that is pertaining to the protein that you want to make, okay? So keep that in mind. We're not copying the entire DNA molecule because that would be wasteful. If we don't need to make keratin, we only need to make hemoglobin, then we're only going to look at the part of the DNA that pertains to hemoglobin, all right? So take a moment now, pause the video, and get this DNA molecule done. Or drawn. So we've got 3' here, 5' there, 5 here, 3'. Okay, so hopefully you had enough time to do that as you pause the video. So what we're looking at here is a DNA molecule. Let's pretend we've got the hydrogen bonding in here. Okay, for an instant in time, we've got the hydrogen bonding going on. And then our bodies say, okay, we need to make hemoglobin. So we need to open up this section of the DNA molecule. In reality, you'd have lots and lots, 600 to 6,000 base pairs, okay? So what we do is we always read 3' to 5', okay? We're in extraordinary circumstances right now, so I'm not gonna make things tricky for you. I will always have the 3' on top and the 5', the strand that begins with the 5' on bottom, all right? So RNA polymerase comes in and it's looking for the TATA box, okay? So I said to you, this is when you have initiation, is when we spy that TATA box. So what are we doing right now? This is called protein synthesis, all right? And we are in transcription. Pardon my arm, it'll be out of the way in a minute. So you're looking at transcription right now, okay? So we find the TATA box, RNA polymerase comes in and finds the TATA box, and then it says, okay, after that TATA box, this is where I now have to start making an mRNA molecule. So using my broken ruler, I'm going to start to assemble my mRNA, okay? So we know that this is the 3' end of DNA, so we always create in the 5' to 3' direction. All right, so make sure that you label that. So after the TATA box, we've got RNA polymerase coming in, and it's going to start to copy immediately after. All right, and it's going to make a complement. So a few things you need to know. This strand here, this DNA strand, is called the template strand. Okay, and it's our template. It's the one we're going to be using in order to make the protein. That's our template strand, okay? This one here is called our coding strand. All right, and even though it's called the coding strand, you would think that we're using it to code, but we're not. This is your mRNA molecule, okay? And I'm going to call it pre-mRNA because it's not ready to leave the nucleus yet. We've got to have some things happen to it before it's ready to leave the nucleus. So here is our mRNA molecule. This is what we're making. And as we go along, I'm going to move rather quickly because I know you can pause the video, and I don't know how much time I'm going to have on this phone before it cuts me off. So what we're going to do is we're going to make our mRNA strand. How? We're going to look at this base here, which is thymine, and we're going to say, okay, thymine partners with adenine, and that's what we're going to put there. Now, what's the partner for adenine? It's thymine, except we're on an RNA. So RNA doesn't have thymine, it has uracil. And so we have to realize every time we have an A, the partner is going to be U. Okay, I was just thinking ahead here with my G. So write your complementary base pairs in there. Okay, and you can check for your accuracy with mine. And if I make a mistake, unfortunately, I don't have anybody here to correct me. Okay, so this is our pre-mRNA molecule. We have gone through looking through the promoter. Okay, there's the promoter. So we have initiation as soon as we start to put this together. Okay, elongation has happened because we have elongated our mRNA molecule. All right, and once we get to a series, I told you, of a whole bunch of A's, now probably better to put one more in there because if not, it looks like a codon. We've got a whole bunch of A's, okay, and we're out of room, obviously. So when that happens, when you get these U's, that tells us that we have termination. So in other words, we're not copying anymore after this point. We're done, okay? Just as a point, if I had the board in front of me, I would tell you this is all occurring in the nucleus. So everything I'm showing you right now, today, is happening in the nucleus, okay? And I just wrote nucleus over there. All right, so this is all occurring in the nucleus. Now, we have termination because we have a whole bunch of A's, and now we've got our U's transcribed there. What happens is that you get some proteins, okay? They bind to that, and they say, that's the end. We're not transcribing any further. All right? So nuclear proteins bind to our series of U's and stop transcription. When that happens, the DNA molecule is allowed to go back together, and we have an mRNA molecule. Okay, so this is our mRNA. Now, again, I don't know how much time I have on this video, so what I'm going to do with you now is just tell you the modifications that happen. All right, so I'm not going to rewrite everything in here. This is our mRNA molecule. In the next video, you'll see this rewritten. We're going to stick with this one. All right. This will indicate the same one, and I will just start it with A, U, G, and I will end it with U, A, A. Okay, so now this is our pre-mRNA. Why am I calling it pre? Because it's not ready to leave the nucleus. If this left the nucleus, it would be taken apart and recycled for its parts. It's because I told you DNA is very stable, RNA is not as stable. So we can take it apart rather easily. So what we need to do is we need to give our cytosol and the proteins in our cytosol some sign that we're not done with this. We're just beginning with this strand. And so what we do, one thing that we do is we put 7 G's at the 5' end. Okay, so we make a 7G cap. It's called capping and tailing. So 1, 2, 3, 4, 5, 6. I need one more G. Okay, so trust me, I've got 7 G's over here. And this is called our 7G cap. So when I refer to capping and tailing, that's the cap. Okay, then we have a poly A tail. Okay, and that poly A tail occurs on the 3' end. So poly A could be many, hundreds of A's. All right, hundreds of them. But we're not going to draw hundreds because we don't have the space, clearly. All right, and so this is our poly A tail. And so one thing that we need to do, these are called post-transcriptional, after transcription, post-transcriptional modifications. Okay, so that's a post-transcriptional modification. What we're going to do is cap and tail. Okay, and that indicates to things outside of the nucleus that this is an important strand of RNA and that we're not ready to take it apart yet. Okay, now I am going to give you quick information here. Normally I would take a little bit more time. There are sections of your mRNA that are called non-coding regions. We're going to deal with those when we get through all of protein synthesis. Okay, so that's days from now. But basically, if I were to draw this, okay, redraw this with no letters in it. Okay, so just imagine that this is what I've drawn. This is my mRNA molecule. Okay, so not to confuse you, I'm drawing this in this form, okay? Because I need it to look like a box. And in this box, we have sections that we call introns, and we have sections that we call exons. And it does not matter how many of each you put in here, okay? So I'm just making these up totally randomly, and et cetera, okay? So I'll make these 2 exons. And so E stands for exon, and I stands for intron. Okay? And so introns are non-coding sections, all right? And they serve a huge purpose, but it's not something we're going to talk about today. Their purpose is not served in protein synthesis. So what we do then is we splice out the introns, all right? So we cut these guys out, and they're gone. And then these exons are left, and the exons are allowed to leave. So this is our pre-mRNA molecule. Imagine we'd have lots and lots of base pairs, not just the ones I've given you here, okay, up here, many more. And so we cut out whole regions called introns, okay? And that is called splicing, and it is done with a spliceosome. So I will get you to read about that. The spliceosomes are on page 322 in your textbook, all right? And the spliceosomes are basically a combination of a molecule with a whole bunch of proteins. So if we look at page 322 for a moment, this is your active spliceosome, okay? And you've got these proteins, small ribonucleoproteins called SNRPs, okay? That's how we pronounce that, SNRPs. And what they do is they actually grip that mRNA molecule and they cut out those introns, okay? So page 322. When these introns are cut out, you're left with your exons, and then your exons can then be reordered in a form called alternative splicing. And I'm almost done with transcription today. Alternative splicing, okay? So alternative splicing means this. If this is exon number 1, and this is exon number 2, and this is exon number 3, 4, 5, 6. Let's say we've copied these, okay? And we want to have, we want to make hemoglobin. And hemoglobin goes 1, 2, 3, 4, 5, 6 in that order, okay? But then we say, okay, you know, we actually have a lot of hemoglobin right now. We've made a lot. Why don't we rearrange these into another numerical sequence to make another protein that we're growing short on? So the amazing thing is, even though you've got these guys here that pertain to hemoglobin, you can alternatively change the numeric order and make a whole other protein, okay? So in other words, if hemoglobin goes 1, 2, 3, 4, 5, 6, okay? Maybe something really similar like myoglobin goes 3, 4, 1, 2, 6, 5. And so we reorder those exons and make a whole other protein, okay? So those are our post-transcriptional modifications. We have gone through transcription. We've looked at initiation. We look for a promoter. We've elongated, okay? And we terminated. So all of those things happened. And then we had to make post-transcriptional modifications, which also occur in the nucleus. And we cut out introns. We left exons in, we added a 7G cap, and we added a 3' poly A tail, all right? And so now this molecule is ready to leave the nucleus.